Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode is focused on the upcoming special issue of Molecular Therapy titled The Biological Clock of Aging and the Possibility of Lifespan Extension. Listen in on the fascinating conversation between Molecular Therapy's Editor-in-Chief, Dr. Joseph Glorioso, and the guest editors of the special issue, Dr. Christin Glorioso from NeuroAge Therapeutics and Dr. John Bell from the University of Ottawa. But first, registration is now open for Breakthroughs in Cell and Gene Therapy, ASGCT's midpoint conference that offers the same impact as the ASGCT annual meeting, but on a smaller, more intimate scale. Join us in Nashville, November 12th and 13th for our most expansive program yet, with 3 concurrent tracks designed for scientists, clinicians, and industry leaders. Abstract submissions for this event are due Friday, August 28th. Visit asgct.org/breakthroughs to learn more. And now, on to our episode.
Hi, everybody. I'm really happy to be here today with Christin Glorioso and John Bell. This podcast is going to delve into some really exciting material, which has to do with the aging clock. What is that, and whether lifespan can be extended. And there's a whole field that is rapidly developing in aging research, and folks are trying to understand what is the basis for lifespan, and what are the markers that determine how long an individual will live, and it varies tremendously among different species. There are a lot of reasons that have been touted for this aging, and they involve changes in the body that involve, for example, inflammation and stem cell senescence, and many others. And so what we'll try to do today is cover some of the main ideas. And just to introduce you, we have a new journal coming out, a special journal on aging, the aging clock, and the possibility of the extension of lifespan. And we've recruited a number of interesting people to talk about their research, and we encourage you to write in and suggest topics that you would like to write about. So let me just jump in here and turn it over to the experts. And I will tell you, Christin, that maybe you could tell us a little bit about the history of aging research and kind of where you think we are today.
Yeah, sure. So I'm Dr. Christin Glorioso. I'm a neuroscientist and a physician by training. And I've had the privilege of being in the longevity and aging field for the last 20 years. And it's really evolved rapidly. So I think it's starting to come into the public zeitgeist just in the last maybe 10 years, with Google starting a moonshot company called Calico, and with Altos Labs and with some other big folks that people know that are celebrities, bringing longevity into the mainstream. But the research has been going on since before the 1960s. So people have started trying to see whether they could extend animal lifespan. So some of the first long-lived animal models were dwarf and Snell mice in the 1960s. So these had growth hormone mutations. And so they were long-lived and small, and they lived about 40 to 50% longer. And then in the 80s and early 90s, a lot of the seminal work was in C. elegans. So people did screened mutations broadly across the C. elegans genome to look to see what genes would extend worm lifespan. And many of those genes related to insulin IGF-1 signaling. So we saw this connection to metabolism. And of course, caloric restriction is one of the most consistent ways to extend life and lower organism lifespan. So those were some of the early ideas. And then people started having that move up the phylogenetic tree and to see what could potentially help with human lifespan, which is kind of where we are today. And now we have longevity biotech companies that are trying to extend human health span across many different disease indications with the thinking that those drugs would also be extenders of healthy human lifespan. And so we're starting to see some of those drugs enter the clinics. Many people think GLP-1 drugs, so Ozempic and Wegovy might be the first longevity drugs that we've seen, and there are many more in the pipeline. So maybe I'll just pause there, sort of high level overview.
That's great. Thank you so much. I was wondering if you could comment on animal lifespan, because there's been a vast difference. And what do you think about that?
Yeah, so we know there are some animals that are very long-lived or even essentially immortal. So one of the most interesting examples, I think, is the immortal jellyfish. So instead of dying, it reverts back to an embryonic polyp state. So it takes its cells and transdifferentiates themselves using many of the jellyfish equivalent to the Yamanaka factors, which are the four factors known to be able to revert any cell in the human body back to a stem cell. That was a Nobel Prize by Shin Yamanaka earlier this century. And those same genes can take adult jellyfish cells and differentiate them into juvenile polyp cells. So they essentially become a baby again and then grow back into the adult state. So if they have, an injury or if they're just becoming too old to be functioning well, they'll rebirth themselves. So they can live indefinitely if no predators eat them or other accidents happen. So that's pretty amazing. And it also may extend to some strategies that people are doing right now in terms of pharma. So there's one of the companies that's writing a review for this issue is working on epigenetic reprogramming, which is using those same factors in the human eye to try and make eye cells younger and treat age-related eye conditions. So this entering phase 1 now in clinical trials. So I'm really excited about that. So that's one example. And then there are tree species that live 14,000 years. The bowhead whales live 200 years. There are some sea grasses that can live many thousands of years. So we know that there are species that are very long lived. I think one of our favorite examples also are tardigrades, which are these little water bear creatures. And they have some things in common. So a lot of them are very resistant to DNA damage. So they have extra copies of repair genes like P53 that can repair damage very well. Other strategies, they renew themselves from stem cells. So the seagrass constantly has a stem cell base where it's growing new grass leaves, but the base is all stem cells. So there are various strategies that animals employ.
That's fascinating. I think there's some sharks that live a very long time, like a couple of centuries or something like that, right?
Hmm.
Yeah.
I looked at the list of people we have on this issue, and it's amazing. I would say, congratulations, Christian, on organizing this because it's really fascinating spectrum of topics. I think that people are really going to enjoy. I'm not an aging specialist. I'm a cancer guy like Joe, and what I found really fascinating about it was, I trained as a virologist and immunologist. I think you did as well, Joe, and then I moved into the cancer space. And there's a lot of overlap between cancer and virology and immunology, and I think exactly the same here with aging. There's a lot of overlap between aging research and cancer biology and immunology. So it's really an interesting field that I hadn't really appreciated before. So I think this is going to be a fun issue.
I think that one of the interesting things is the notion of cellular senescence, and some cells in the body reproduce or quite often, like skin cells or cells in the intestinal tract. And this senescent cell idea should be, I think it's monitored by the immune system. And so you have cells that are stem cells, but they no longer, can develop into mature cells. And sometimes when they do, they acquire mutations that make those into cancer cells. So it's a very interesting problem in terms of senescence. So senescence happens both at the cellular level and the whole organism level. And I think that all these systems talk to each other. So it's going to be fascinating to try to figure this out.
Would it be fair to say, Kristen, that, well, one theme I've seen through some of the articles that you're recruiting to this is the concept of DNA repair. In fact, I think in the case of, is it the case of the bowhead whale that the DNA repair systems are ramped up or something? Is that my understanding?
Yeah, there's several species that is thought to be the mechanism of them living longer. So bowhead whales, tardigrades, naked mole rats, which are rodents that normally mice live 2 to 4 years, and these naked mole rats live 20 years. So this tenfold lifespan extension of the same genus as the mice, and that's all thought to be mostly DNA repair mechanisms. So that's a huge factor. But I think there's other ones as well, regenerative capacity and
Is, is this due to gene duplication of repair enzymes, do you think, or are they protected in some way? I mean,
so some of the species I know have extra copies of P53, for instance. And then other species have different copies of the DNA repair machinery. So there's some mutations in them or changes in their function that allow them to function better. And I think you and I were talking recently about how, why would you ever not want to have good DNA repair? And I think that's kind of an open question. But, one thought was, if aging is programmed, that's the one mechanism of making sure we have a lifespan that makes sense for our species by limiting it with poorer DNA repair than some of the other organisms. So,
yeah, I think we
yeah.
also discussed the possibility that the lifespan was an adaptive process. That is, evolution had limited the lifespan of some animals because they needed to be more adaptive to the environment. So I think that's a possibility. So maybe, Kristen, you could talk a little bit about aging brain problems, because this is something that is very on everybody's mind. I don't want my brain to disappear where I can't reason anymore. And so diseases like Alzheimer's or Parkinson's disease, which are multifactorial problems, they are, those are on everybody's mind to try to figure out how to solve dementia. Maybe you could comment about that.
Yeah, so it's a problem that's pretty near and dear to my heart. Alzheimer's runs in our family, and I myself have an ApoE4 allele, which is the most common genetic risk factor for Alzheimer's, puts me about 2 to 3 times the risk as the general population. People with two copies are 10 or 11 times the risk, depending on what other genetic variants they have. And so the question has been, what causes neurodegenerative diseases? I think we're still figuring that out, but we have a lot more information than we did 40 or 50 years ago. All neurodegenerative diseases are different flavors of the same problem. So they're death and dysfunction of neurons in particular parts of the brain. So if the neurons die in the hippocampus, that causes Alzheimer's. If they die in the substantia nigra, that causes Parkinson's. If they die in frontal and temporal regions, that causes frontotemporal dementia, and so on. And so there's also different pathologies that we see with these diseases. Alzheimer's is the most common form of dementia, so I can speak to that specifically. What we see are toxic proteins that build up in the brain, amyloid plaques and tau tangles. And people with with ApoE4 alleles like myself are more high risk for having this amyloid build up. And so many of the pharma companies have, for the last 30 years, had a hypothesis called the amyloid hypothesis, which is if you can just remove these toxic proteins that builds up, you can cure Alzheimer's. And so there were billions of dollars of failed clinical trials. So that hypothesis didn't work out for about 20 or 30 years. And there's also some counter evidence. So people that get brain scans and have a high level of amyloid plaques in their brains, not all of them have memory problems, and not all of them will go on to develop Alzheimer's. They're more likely to develop Alzheimer's, but certainly there's people with tons of amyloid plaques that don't ever develop Alzheimer's. And so the story has gotten kind of interesting. There's been a couple of antibody therapies that have been approved in the last 5 years that remove these amyloid plaques with the thinking, okay, if we remove the plaques, that should cure the disease. And what happened is they work a little bit, so they slow progression by about 20%, but they do not cure Alzheimer's and they don't work for everyone. And so the way that I'm interpreting these results is that amyloid is one risk factor amongst many, and that it's perhaps one piece of that puzzle, but there's many other pathways that we need to target, particularly aging pathways. And so what I've studied for most of my career is what happens in the aging brain. And so what we see is even starting in your 20s, there's loss of synapses, there's DNA damage, there's mitochondrial dysfunction, there's all of these other pathways that are happening that are huge risk factors for cell death and for Alzheimer's progression. And so we're going to see drugs that are targeting the whole program of aging, not just these toxic proteins.
It's an interesting point, Christine, because that's another overlap between the cancer world and aging, is that it's multifactorial, that there are risk factors, but none are 100%. And so what I like about the issue you've done here is we're really looking at things like protein translation, metabolism, DNA repair, like quite disparate types of biological processes, which all can impact aging, as you said.
Mm-hmm.
Christin, what do you think about the connection between epigenetic changes and, say, mitochondrial function?
Yeah, so people have been studying changes in methylation, which affect gene expression for a long time, and what has been pretty popularized over the last 15 years are what is known as aging clocks. So you can look at methylation marks on the DNA, and those correlate with chronological age, and your clock based on how many marks are in the up or down position on the DNA can actually predict disease risk. And so people think that these epigenetic marks are controlling gene expression, which is what's controlling the rate of aging. So if you think of aging as a program that's controlled by transcription factors like the Yamanaka factors, part of how they're working is through these epigenetic programs. And so how that relates to mitochondria, which is your original question, I think is still being elucidated. So how these transcription factors themselves are then impacting mitochondrial health or biogenesis, or getting rid of mitochondria that have mutations or older mitochondria. I think we're still trying to understand how those two things connect.
How important do you think inflammation is in aging brain problems?
I think it's hugely important. So we know that people that have neurodegenerative diseases have a breakdown of the blood-brain barrier, and that means that there's more inflammation going on in the brain. People have also pointed towards metabolic disturbances that can increase inflammation. We know things like white matter hyperintensities are increased. Those are inflammatory cardiovascular problems in the brain. And so I think it plays a big role. There's a company called BioAge Labs that's actually going into phase 2 for NLRP3 inhibitor, and NLRP3 is part of the inflammasome. And so that's thought to be a longevity pathway in general. Inflammaging is one of the 12 hallmarks of aging, and we think it's particularly important for the brain. And so we're going to see brain penetrant versions of these NLRP3 inhibitors that take a crack at Alzheimer's. coming soon. I think that the next 5 years we'll see clinical trials for those in the brain.
That's
Yeah.
fascinating. What are some of the other factors that contribute to aging? For example, lack of sleep or lack of exercise or our diet, for example. How are those ranked these days about what impact they have on aging?
So speaking to the aging brain, I would say sleep, diet, and exercise are one, two, and three, in terms of having the biggest impact on how fast or slow people age, how many age-related diseases they have. So there's a lot of evidence for the Mediterranean diet. We know 10,000 steps a day is very protective in a number of clinical studies, particularly cardio. So people getting their heart rate up into zone 3 or zone 4 is really great for the aging brain. Sleep, the ideal sleep is 7 to 8 hours. Less sleep or more sleep is both associated with worse outcomes. So those I think are the big three, but there's many other things that factor in: stress management, making sure that you get outside in nature. Community is a huge one. So we know that the number of friends you have can actually predict your lifespan. So that social connectivity piece
Yeah, that's
is really big. Yeah.
What about these blue zones that exist? Well, people talk about blue zones, and I guess that's a combination of things like you just spoke about, stress and diet and so on. Is there a consensus on why those blue zones exist yet? Or?
Yeah, I think people are trying to figure that out and figure out what they have in common. So we know that they tend to be coastal communities that walk a lot. Okinawa, Japan, Sardinia, coastal Costa Rica. And so these are some of them. There's even one in California, Loma Linda, California. It's the only one in the US. And so we know that people tend to walk a lot. They tend to have a lot of community. There's a Mediterranean style diet in these places. So these are some of the commonalities, but it's certainly a combination of different factors. I think the
Yeah.
coastal might be a seafood that may be an omega-3 connection, with
Mm.
seafood eaters.
Mm.
So, what are the sort of standard things that limit lifespan? Obviously, disease of various kinds and fatty diets and smoking and things like that. But if we fix all of that, I doubt that at least currently people are going to live that much longer. They may live longer disease free, but extending the lifespan is another issue, and I think that it's going to be fascinating to see if we can crack this.
Christine, as you're an aging researcher, it must be really an exciting time because all these tools we have for studying biology, molecular tools that we're really implementing in the cancer space and other spaces are at your disposal now. And that must be really changing how we think about the aging process because we really look at this at the molecular level now.
Yeah, absolutely. And I think we're going to see, certainly the genetic approaches with gene therapies and what we've learned about the pathways. We're going to see show up in some of the clinical strategies for sure. So, but I do think there's currently it's thought that there's a hard limit because we've extended median lifespan a lot. So, on average, people are living much longer, but the maximum lifespan has stayed pretty consistent. So, Jeanne Calment is the longest lived person ever. She's a French woman who died at 122. And so we haven't had people live much longer than that. And, I think we're going to be able to treat all of these age-related diseases. I mean, I think neurodegeneration is part of it. We haven't really made much headway with that, and that jumps from the fifth leading cause of death to, the top one of the top two once you pass 105 or so, right? So that ends up being the biggest problem when you really get up there in age. But I think there may just be a hard limit, even if we cure all cancers, even if we have perfectly working hearts, which are one and two in terms of what kills people. And then we're going to need replacement strategies. So that's sort of the next frontier is how do we replace enough body parts to live longer? And there are people that are working on that as well. So
We need to
yeah,
be like lizards and grow a new tail, right?
yeah,
Uh, so are any biotech companies working on regeneration specifically, or do you know about like make
yeah.
stem cell
So
replacement, that kind of thing?
yeah, there are a lot of companies working on that. So, at least in Parkinson's in Japan, I think we have the first approved stem cell therapies for injecting stem cells into the aging brain to treat Parkinson's. So, that's been approved in Japan and is probably coming here. And there are a lot of other companies working on rejuvenating other organs. So one of the people writing for the review is a company called Xtherma. So they're working on preserving organs for longer. So part of the organ transplant problem is that organs will only last for a short amount of time, 24 hours, and then they'll go bad if you can't get them to the right match in time. And so if you can just extend how long with better freezing techniques and better preservatives, how long those organs last, then you can get them to more people. And that, also the really long-term implications of that are freezing whole people. So like cryopreservation
Hmm.
so if you have some fatal disease that we might be able to cure in 30 or 40 years, we'll just freeze you down and warm you back up once you have the cure to the disease.
And hope you're not mush, right? Well, I think there's a lot of interest in organ cultures and all that. So I think probably, I guess one of the things we could try in the future is growing whole organs or parts of organs in a petri dish and then use those for transplant. Is anybody trying that?
Oh yeah, there's a lot of people working on that. I think George Church's lab at Harvard has a big program around that. A lot of people are using organoids for testing drugs right now, but if those get good enough, those can just be transplanted. And then there's some even like pretty far out companies in San Francisco where people are trying to grow babies in petri dishes or clones of people without brains in petri dishes to transplant into people. So
Yeah.
yeah.
One of the Star Treks, they tried to transplant the brain. Remember that? Anyway, so one of the other things I think is thinking about the future is that there are a lot of diseases, th probably 5,000 genetic diseases that probably with some kind of really perfect CRISPR situations, we could edit the human genome in ways that we could eliminate a lot of these diseases, and then they'd be inherited. So that seems like that's an inevitability because otherwise you have to. The only thing we can do is gene therapy later or other kinds of treatments, including transplants. So I think that some of the obvious targets are mutations that cause cancer. So I think this is something that I don't think the population is ready for, but into the future, I would guess that this is going to become something real. And the other thing is that trying to understand differentiation of stem cells and how they're, what are the genes that target, say, a cell to become this kind of cell or that kind of cell. And that requires a really deep understanding of the pathways involved. So I guess that proteomics is going to become a really important field to understand how differentiation occurs.
Yeah, for
Yeah,
sure.
and to that point, Kristen, is there a lot of work in the proteomic, like doing the proteome of someone like my old now, and compared to you, do we have different proteomes, different genes expressed, or is the dark genome involved in any of this too? That's sort of the new theme, is that dark genomes may be activated in cancer. Is that something that's happening in aging as well?
Can you explain the dark genome? I actually don't know about this.
Oh well, it's just there's some recent papers about how there's a lot of peptides and proteins that are actually thought were not thought known to exist. They're in the non-coding regions of the DNA, but they've recently been shown to be expressed. And in cancer, you often see activation of these parts of the genome, and it's
Mhm.
thought that maybe they influence inflammation and so on. But I wonder if that's also maybe an area that we could think about for research and the
Absolutely.
agents of.
So yeah, I mean, I think about it a lot in terms of diagnostics. So, what the public is currently has in terms of tracking their own health are, standard biomarkers like cholesterol testing and ApoB testing and glucose. But there's a whole wave of new diagnostics that are coming out that are so, at my company, NeuroAge, we're looking at whole genome blood RNA-seq, for instance. So RNA sequencing, so the entire transcriptome. And then there are other companies that are doing whole genome proteomics as well that are brand new companies. There's going to be a generation of blood diagnostics that are now that proteomics is becoming cheap enough and they're still working on how to get this to people in their houses. So currently it has to be cold. And so there's some little logistical things to work out that are almost solved for. But we're going to see a whole slew of diagnostics where people just have their whole genome, everything on many different levels. And that's going to tell us, what diseases you're at risk for, what pathways are involved. Like if you're at risk for Alzheimer's, why? Or if you're at risk for breast cancer, why? What's driving that? And people are going to have access to that kind of data in real time, I think. So I think that's going to be very exciting. And the dark genome is even one step further. So I think people are not even looking to see, what we didn't know about yet, but that's also going to be probably coming out of these rich data sets. The first examples we have of commercial companies, I think, are companies like Grail from Gallery, right? Gallery from Grail, which is cancer screening through epigenetic testing that is on the market now, and companies doing microbiome through stool RNA and DNA testing. And so we have kind of the first examples of these, but I think, we're all going to know what's happening with our proteome in real time every day in the future, which is going to be wild. Yeah.
Well, I think there's so much interest in the microbiome and its connection to the body, and even the brain, of course. And so, are people looking at that in terms of aging models?
Yeah, absolutely. There's mouse experiments where they transplant, a young healthy mouse's like fecal transplant from a young healthy mouse into a like an older obese mouse, and the older obese mouse becomes lean again and healthier and has less age-related disease. So we know there's this connection between gut microbiome and health of the brain and other organs. And I think we're just beginning to figure out what the proper balance of organisms are and what each does and how to influence that. So there's a bunch of commercial testing companies. I haven't tried them myself yet. It's on my list of biohacking things I want to do is to test my microbiome.
Biohacking. So I guess the last question I really want to ask is, what do you see in the future? I mean, do you really believe that we can extend lifespan, and what do you think has to happen in order to pull that off? That's an easy question, right?
Yeah, so John, do you want to go first? Do you want to take a crack?
We definitely can do it, Joe. Don't worry about it. Yeah, I know you and I will. It's all going to happen. I mean, I think there's already there's stuff that Christin sent. There's already some, at least in vitro data, that you can manipulate genomes and extend lifespan to some extent in vitro. So I think it's possible once we understand, and maybe it's actually the application of AI to really start to decipher what all these various factors are. But I think it's going to be possible that we could extend lifespan.
Yeah, I
Yeah.
think like the GLP-1 story is really interesting because it's an agonist, and it's a longer lasting protein that has been engineered, and it has a fatty acid attached to it, so it binds to fibrinogen, or that's not the right one. I can't remember exactly what it is, but its life in the body is days as opposed to minutes. And so engineering peptide hormones, I think, is going to be a really exciting area, and maybe will be important to managing the aging process.
Yeah, absolutely. And GLP-1s actually, at least in animals, they work through AGE1 or PI3 kinase pathway, which is a known aging pathway. It's one of the experiments from the 80s and 90s when they could extend worm lifespan. One of the most famous genes is AGE1. And so GLP-1s work through that pathway. And in mice, at least, they protect cells in pancreatic islet cells, so cells in the pancreas that produce insulin from dying, and they also protect neurons from dying. So there's some evidence that they're not just affecting people's weight, they're actually working through longevity pathways, which is pretty cool. But I think, what people can do now is optimize their lifestyle. So we know that if you do all of that, you can probably add another 10 years of healthy life. And maybe that will buy you some more time for the exponentially accelerating, cool, bigger tools that are coming down the pike. I think the first tools are going to be things like the GLP-1. So we'll see these longevity drugs that affect known pathways. So we'll optimize, everybody will have great inflammatory profiles and clean arteries. And so we'll start to fix all little
Yeah,
issues that we have combined with the diagnostics. And then we're going to end up with the heavier guns, which is like replacing all your organs and growing things back for you and giving you new stem cells parts.
yeah.
And eventually we're going to become robots with upgraded.
That's so true. I was just reading a book about that actually, where they take a
That's one
consciousness
way to
from
survive, is you're a machine rather than a person.
exactly, yeah.
Okay, well, I don't want to drag this on too long, but I think it's been a really fascinating discussion, and I'm really enthusiastic about aging research, and I knew very little about it until you kind of enlightened me, Kristen, and I've just been
Yeah.
reading about it, and it's a very exciting field, and I'm hoping that some of the you know s- some of the special things that whales can, or not whales, but sharks can live a long time because they have better DNA repair, or tardigrades or whatever. But we could probably engineer the human genome to have those changes, and maybe that'll be something we would try in the future. Who knows?
Well, that's my question to John. What do you think if we all had perfect shark copies of P53? Do you think that would work for us as a cancer biologist?
Yeah, I'm going to start a company on that actually. But I, I think it's known, right, that isn't it elephants that have multiple copies of PP3 and they have very long ones. So there's starting to be biological evidence. I think that in the case of the bowhead whale, it's an enzyme that we all have, but it's expressed at a much higher level, I think, if I recall. So,
Yeah,
there's the potential there to make this happen.
right.
It'd be really interesting.
Thank you so much for participating in this podcast. It's been really interesting and a lot of fun. So I think we'll sign off now.
Yeah. Thank you so much. I really enjoyed it.
Yeah, thanks, guys. Really appreciate it.